subscribers, welcome to today's session. So children, I hope you have really enjoyed your Sunday, but now get back to studies. Today we are going to discuss about the classification, of course the fungi. We have discussed about the monerans, we have discussed about the protistans, and today we are going to start our third kingdom, which is the fungi. So why fungi was not kept in monera or either in the protista, nor in the animal kingdom or in the plant kingdom. It has its own individual kingdom named after it called kingdom fungi, and we are going to discuss all the classification and related characteristics, their habitat in today's session. Okay, so children, before we start, let me introduce you to myself. I am Anam Khan. I'll be your biology expert for the next session 2023 and 2024. We're going to discuss all the amazing and interesting topics related to biology/botany. Okay, coming back to our today's session, let's start with the fungi. So children, talking about the fungi, we know before we start the fungi, these are all the particular topics which will be covered in the coming sessions. We have already discussed the biological classification, why it was necessary, how exactly 2 kingdom, 3 kingdom, 4 kingdom classification came into the limelight by different scientists, and why every particular classification has its own drawbacks. Coming back to, we have already discussed the features of monerans in the kingdom monera. We have seen how exactly the basic characteristics they possess, their nutrition capacities and how they reproduce and their cycles. Yes, and we have also discussed about the kingdom protista, their characteristics and why they were not kept in either monerans or in the plant and animal kingdom, which unique characteristics they possess. And today we are going to discuss about another kingdom, that is the kingdom fungi, and the coming session we are going to discuss about the kingdom plantae. Of course, algae, bryophytes, pteridophytes, angiosperms are yet to be discussed. We will start with the chapter kingdom plantae or the plant kingdom. There we are going to study over there, and of course the viruses, virus, prions and lichens are also part of the whole biological classification. Okay. In the today's session, we are going to discuss about whole kingdom fungi. We are going to discuss about the hypha, aseptate hypha, septate hypha, the whole mycelium, nutrition in fungi, and of course, we are going to discuss about the basic classification of fungi, how it is being divided into whole subtypes, subclasses, and what are the features each and every one of them possess. Okay, so let's start our today's session talking about the fungi, talking about the basic characteristics. Before we start, so in the previous session what we have studied, let's have a quick recap of whatever we have studied about the protista. So children, you know, in the protista we have gone through about the slime molds. So slime molds are typically fungi-like protistans. They feed on the dead and decaying organic matter. The plasmodial stage resembles the protozoa, and the fruiting bodies form spores and resembling fungi. The spores have a cell wall made up of cellulose, and they are resistant to adverse condition. They can survive up to many years. Okay. Next, the acellular slime molds feeding stage is multinucleate mass of protoplasm, that is plasmodium. Of course, there is a whole plasmodium, whole life cycle of the slime molds we have already discussed, and we have seen exactly how different acellular, how they basically have this layer during the unfavorable conditions, and how exactly they survive and how they basically produce new organisms. Okay, how they reproduce. Cellular slime mold's feeding stage is a single-celled amoeboid which lives as a solitary organisms. Okay, so basically they also live as a solitary organisms. They have features like the fungi. They are typically saprophytic. They have their own individual method of surviving in the adverse condition, which is the unfavorable condition, by forming a plasmodium. This is all we have studied in the previous session. Coming to our today's session talking about the fungi, let's see what are the characteristics that the fungi process. So children, talking about the fungi's characteristics, it is the kingdom fungi is enormous with 1 lakh different species, 1 lakh to 10 lakh different species, and they are completely saprophytic and parasitic in nature. It means they have this saprophyte, they feed on the dead and decaying animals or the organic matter, and they also show a parasitic nature. It means they have a host body to live on. Okay, these are the main decomposers and essential for the recycling of inorganic resources. Yes, combining with the bacteria also. When fungi and bacteria feed or basically decompose, okay, so they are decomposing a lot of inorganic matter. So yes, majorly the fungi, which is a typically saprophytic, feeds on the dead and decaying organic matter. That is what, by basically degrading, okay, or basically what you say, recycling a lot of inorganic resources. Okay, now few fungi are also living as a symbionts. What are symbionts, children? Having this mutual relationship with each other, benefiting from each other. Okay, some benefits are fungi is giving an association with the algae, okay, which is the lichen, which is an association with algae plus fungi. Okay, so basically the algae plus fungi is a lichen, then there is a mycorrhiza, which is basically having association with the plant roots. Okay, so what did they do? They having benefited from each other. Basically they are obtaining benefits from each other and they are living like a symbionts, like a mutual relationship. Okay, and next, the study of fungi is known as mycology. If you are going to study about the detailed description or detailed study about the fungi, that study will be called as mycology. Okay, so children, this is the very basic characteristics of the fungi, that what kind of a nature do they possess, how they are basically present, and studying or basically is called as mycology. So these are the few very basic characteristics. Let's see what we have next. Talking about more of the fungi, we have a structure. Yes, they have a very unique structure, children, very unique structure. It means the fungi have a multicellular and a unicellular like yeast. All the fungi are multicellular in nature except the yeast. Yeast is the only fungi which is the unicellular in nature. Otherwise all the multicellular, all the fungi, all the structures are typically multicellular, and the only exception is the yeast. Okay, and what makes the fungi very much different from the plants, animals, monorans, and of course the protista is the cell wall. The whole cell wall, the cell wall of fungi contains chitin or non-cellulosic polysaccharides. Yes, this is a very important point to be noted, that what basically makes a fungi different from the other people or the other, not say other people, other organisms, that we have to have the individual kingdom for fungi is majorly of course also because of the cell wall and of course the nutrition nature, but the major is also the cell wall. The cell wall is constituting, it is made up of chitin and non-cellulosic cell wall. Okay, the most cases the Golgi body is unicisternal. Cisternal means cisterni. These are Golgi bodies, these are cisterni. So in the fungi, they might have a unicisternal, a single cisterni, which is composing of a Golgi body, which is also a structure of the fungi existing in a fungi. They have lomasomes, very important, which is infolding of the plasma membrane to increase the surface area. Yes, the plasma membrane of the fungi have infoldings, and those infoldings, the plasma membrane of fungi has infoldings. What you can also say, these kind of infoldings. So basically what they are called as lomasomes. What they do, they just basically increase the surface area, they increasing the surface area of the fungi. Okay, and the body is made up of a filament which is called hyphae. What is a filament? Simple. This is a spore. If this is a spore, this spore will convert into a filament like structure. This is your filament, and this is what you called as hyphae. If this filament forms a network, if this filament is forming a network, whatever network they are basically creating, whatever network they wanted to create, they can create, and that is what you called as mycelium. That is what you called as mycelium. Okay, so basically a hyphae is a filamentous like structure. Okay, a different type of hyphae is also existing, we will discuss about that, and the whole network of hyphae is basically creating a mycelium. Okay, a network of hyphae is called mycelium. Let's see, also have a look at the video. You can see this is a spore, and this filament like structure which is coming out of a spore is your hyphae. And children, as soon as it grows out, it will form a network. You can also say, see, this is how other filaments are also growing together. They are basically going to create a whole network. This is what you called as an hyphae. The whole network of the hyphae is called mycelium. This is how they are creating the mycelium network. This is what you called mycelium. Can you see this? This is how the fungi grows through a hyphae, creating a network of hyphae which is called a mycelium. I hope this is clear. I hope this is very clear that what is mycelium and what is hyphae. Okay. So talking about the structure, this is how the structure of fungi is maintained. They are unicellular, only the yeast is unicellular, they are multicellular, and they contain a chitin cell wall and non-cellulosic cell wall. In most of the cases, Golgi bodies is unicisternal and they have a hyphae. A network of hyphae is called mycelium, and yet that typically saprophytes, and a body is a filamentous which is called the hyphae. A network of hyphae is called mycelium. The plasma membrane has an infoldings which is called lomasomes, which increases the surface area of the fungi. Clear? So this is about the structure of the fungi. Let's see what we have next. Talking about the mycelium only, children, the habitat. Before we further describe the types of hyphae existing, we are going to see that what kind of a habitat they reside in. So typical when we talk about the habitat, the fungi is a cosmopolitan in occurrence, present everywhere in waste, air, soil, animal, and of course plant. Yes, they are cosmopolitan, they are present everywhere, they are in water. Just leave, don't do anything, just leave your bread on the fridge or maybe inside also. Sometimes it reaches inside the fridge also. Yes or no? If you leave your bread on the fridge or maybe inside a fridge for some days, after it is expiring, you will experience the fungus has taken over the bread. Yes, even it is inside the whole, what you get, poly bag, whatever cover it is basically coming in. You see the fungus has basically taken place, the whole covering of the bread, which is the rhizopus, which is the bread mold, of course we call it. That is how the fungus can reach anywhere. Okay, your skin sometimes has a fungal infection, your dandruff is what is a fungal infection only. Okay, people experience a lot of dandruff on their scalp. That is what? That is fungal infection. Okay, so children, they are typically cosmopolitan, they are present everywhere. Water, air, soil, animal as well as the plant. Talking about when they are inhabiting in dung, they are called as corpophilous. When they are growing on the bark, they are called as corticocollous. When they are growing on the wood, they are called epixylic. And they are growing on the hair, skin and nail, they are known as keratinophilic. So on the basis of the habitat they are residing in, we have categorized them, we have divided them into few, that which particular habitat they live in, they are basically named after that. If they are on the dung, corpophilous. If they are on the bark, they are corticocollous. If they are on the wood, they are epixylic, and if they are on the hair, nail and skin, they are known as keratinophilic. So these are the few places where you can find out the fungi, and basically this is completely cosmopolitan. Okay. So children, this is the basic features of the fungi. Before we get into the deep classification of the fungi, as you can see, this is how this is growing. This is growing on the nails. This is how the fungal infection basically takes place. Keratinoxylate. This is on the bark. Can you see this? This is on the bark. Okay. And yet the other on the dung, we have just now seen the dung, bark, and of course the nail skin, hair or the nails. So these are the few habitats in which you will find the fungi. Okay, clear? Okay, so children, moving forward, talking about more about the fungi nature, let's have a. So children, there are two types of mycelium, septate and aseptate. What is the function between a septate and a aseptate mycelium? Listen very carefully. Can you see this? The two have a very big division. Talking about the mycelium, which is a network of hyphae. Hyphae is a single filamentous like structure. The typical mycelium is further divided into septate hyphae, which is having separation, or non-septate hyphae. So talking about the septate hyphae, they have cross walls or septa. They have cross walls is completely absent. Okay. Talking about the non-septate hyphae, the cross walls is completely absent. It means the division is completely absent. It means having the nucleus freely flowing in the cytoplasm of the hyphae, creating a cenocytic condition. Okay, there is no complete division. As you can see, children, there is no division. They are freely floating inside the cytoplasm. That is what you call a non-septate hyphae. But when you are talking about the septate hyphae, the clear division is there, and each division has its own nucleus, creating a cenocytic condition. What is a cenocytic condition? When the nucleus is flowing in the cytoplasm freely. This is freely flowing nucleus inside the cytoplasm. When we talk about the septate, their cross walls are present, and it is further divided into two types. So children, on the basis of the cross walls, they are further divided into primary mycelium and the secondary mycelium. What is the primary mycelium and what is the secondary mycelium? Talking about the primary mycelium, each septate has nucleus, one nucleus which is called as monokaryotic. Each septate has only one nucleus which is called as monokaryotic. Okay. And secondary mycelium is that each septate has two nucleus, creating a dikaryotic division. It means when the primary mycelium further undergo division, there is a nuclear division, and yet each separation will have two nucleus, creating a dikaryotic stage, and that is what you called as primary and as well as the secondary condition. So secondary mycelium is a condition when there are two nucleus present in one separation, creating a dikaryotic or dikaryon conditions. Okay, children, the part of the mycelium is absolutely important because it is always creating a confusion, because when we are going to study about the classification, we are going to see that different classified fungi in the classification possess different kind of a mycelium, and the kind of mycelium they possess, their reproduction is also depending upon that. Yes, tomorrow we have a class with respect to the reproduction of fungi only, because there are very vast reproduction in the fungi. That is why I have kept an individual session for it. At the same time, that how basically the reproduction and nutrition is taking place in the fungi. Okay, at the same time we are going to discuss that in an individual session. Okay, moving forward, this is a clear classification of the mycelium: septate, non-septate, primary mycelium, secondary mycelium. Okay, moving forward children, talking about the whole mycelium, we have going to talk about the nutrition. So children, as you can see completely here also, the nutrition is typically of three types. They are completely heterotrophic. Fungi are completely heterotrophic, and when we further divide the heterotrophic, it is further divided into three categories. One is the saprophytic, the other is the parasitic, and the third is the symbiotic. The fungi is completely heterotrophic in nature, and we further divide the heterotrophs. They are completely divided into three categories. One is the saprophytic, the other is the parasitic, and the third is the symbiotic nutrition, mode of nutrition. So what is the saprophytic mode of nutrition? This is something which is very prominent, very evident in the fungi, that they basically feed on the dead and decaying organic matter. Okay, yes, you know what is dead decaying organic matter, which is your bread. Of course, the rhizopus, which is the bread mold. They feed on this. This is expired. This is a dead, this is a completely decaying matter. Of course, they feed on this dead and decaying organic matter. So when we talk about the parasitic nature or parasitic mode of nutrition, these fungi gets its nutrition by living on the organisms like plants or animals. You know what are parasites, children? They have to have this host body to live on. Okay, they feed on the host body because not killing them, but just they are feeding on them. Okay, just like even the plants as well as the animals. Example, the Puccinia is the example for the parasitic nature of the fungi or the parasitic mode of nutrition in the fungi. Talking about the third, which is very important and very popular also, which is the symbiotic mode of nutrition. Yes, this is something which I have told you in the initially when we started this session, that symbionts having this mutual relationship, each and both of them are benefited from each other, having this symbiotic relationship, living together and getting benefits from each other, in which the few fungi are found symbiotically associated with other organisms to obtain their nourishment. Example, lichen. Lichen is a symbiotic association with fungi and the algae. This is what I have told you. The fungi, why algae? Because algae does the photosynthesis. Algae is autotrophic in nutrition, while the fungi brings up the moisture, the water it needs. Algae needs water, right? Yes or no? And in turn, what algae gets? Algae gets water also, and algae gets a place to gets home also. It gets a place to resides also. So that is how they are having this mutual relationship. You give me home, I will give you food. Okay. So fungi stored in the form of a glycogen. They store their food, whatever food they are basically having, they store the food in the form of a glycogen. So there are three basic nature or basic mode of nutrition which is taken by the fungi to obtain their nutrition. I just hope this is completely clear. Okay. So we have discussed about the basic features of fungi, we have discussed about the mode of nutrition, and we have also discussed their habitat. Okay, and we have seen when we talked about the structure, the two different type of mycelium are existing, which is the aseptate and the septate, of course, primary and as well as the secondary. Clear? Clear? Okay, so this is all about the nutrition part. Talking about the further fungus, how the classification is done. Yes, this is very important, talking about the classification, children. The whole criteria of the classification of the fungi is on the different types of morphology, reproductive structure, physiology and their life cycle. Okay. Okay, children. So on the basis of their structure, their life cycle, their physiology, their morphology, the fungi are divided like this. Okay. The Phycomycetes or Phycomyta includes two groups, which is Oomycetes and Zygomycetes. Let's not that concentrate over here, let's concentrate over here. Let's see, this is your Eumycotina, which is a true fungi. Just concentrate that this is a true fungi. Then we have divided into two types of fungi. One is the lower two fungi, which is have aseptate mycelium. Aseptate mycelium means no separation. They have called it as lower true fungi. Okay, primitive ones. Then there are advanced fungi which is called as septate mycelium, which is the higher true fungi which have a division in their mycelium. They are more of an advanced kind of fungi. Okay. They further divided into class called oomycetes and zygomycetes. The oomycetes are called water molds, while the zygomycetes are called conjugation fungi. Okay. Talking about the higher fungi, they are classified into sexual reproduction known and sexual reproduction unknown. One who reproduce through sexual reproduction but do not show the sexual reproduction. The further classified into ascomycetes, which is known as sac fungi, and basidiomycetes, which is known as club fungi. Okay. And the only fungi which is not showing the sexual reproduction is the deuteromycetes, which is also known as fungi imperfecti, which is also known as fungi imperfecti. So these are the classification, these are further classified lower fungi, higher fungi, and the typical class is basically they are divided into oomycetes, zygomycetes, ascomycetes, basidiomycetes and deuteromycetes. Each and every organisms residing in that particular class has their own characteristics to possess, and we are going to discuss all of them, do not worry. So this is how the classification of the fungi is being done on the basis of the reproduction morphology. Morphology is of course the mycelium, of course the whole morphology, physiology, and of course the reproduction. This is what I have said, the fungus classification is typically on the basis of morphology, reproductive system, their physiology. Okay, this is how they are being divided. Moving forward, talking about the very first, it is the phycomycetes. What is the phycomycetes? This is what you have done. Phycomycetes, which is further, or phycomyta, which have two group, oomycetes and zygomycetes, which is this. This is the phycomycetes. This is the phycomycetes, which is further divided into oomycetes and zygomycetes. Remember, the phycomycetes is divided into oomycetes and zygomycetes. We are talking about the phycomycetes, which is found in the aquatic habitat. Okay, the phycomycetes is further divided into two, which is zoomycetes and oomycetes. Clear? So children, let's discuss the features of the phycomycetes. Talking about the phycomycetes, they are found in the aquatic habitats, present on the decaying wood, in the moist, as well as the obligate parasites on the plant. Okay, they are the obligate parasites on the plant. The mycelium they have is typically aseptate, and whenever there is an aseptate mycelium, whenever there is no division in the mycelium, you should always consider it is going to be cenocytic. It is going to be cenocytic. Talking about the asexual reproduction, children, it is by the zoospores or phanospores produced in the sporangium. So the sporangium, these are the sporangiums, reproduce spores called zoospores, which is basically will be released and will create a new fungi or the new particular organisms. This is how the asexual reproduction is taking place. So children, how the zygospore is formed? They also show sexual reproduction in which the zygospore is formed by the fusion of two gametes. Yes, they also show the sexual reproduction through the formation of a zygospore, and the zygospore is formed by the fusion of gametes. What gametes? Gametes are isogamous. Isogamous, they have same structure. Iso means same morphology, same structure, and anisogynous or oogamous. The gametes can be same structure, same morphology, they can be of similar morphology, and they are completely different from each other, yet they are showing the fusion. The example of the phycomycetes is the mucor, the rhizopus and the albugo. So these are the few characteristics of the phycomycetes. They are aquatic in nature, they say sexual reproduction through isogamy or isogamous and anisogynous or oogamous. They have the fusion of gametes by showing the sexual reproduction. They can also reproduce asexually by forming a spore through the sporangium, and the basically phycomycetes are further, the examples of the phycomycetes are the mucor, rhizopus and albugo. Please remember, they are typically obligate parasites on the plants. They cannot survive. Moving forward, discussing about the phycomycetes, next we have the sac fungi. which is the ascomycetes. Children, the name itself is kind of a very clear, sac fungi. Okay. So, talking about the sac fungi, children, the sac fungi is mostly multicellular penicillium unicellular yeast. Children, I have told you in the initial also, so the fungi has a multicellular structure, but the only unicellular fungi is the yeast, which comes in the class of ascomycetes. Ascomycetes is yeast. Okay, is the only fungi which shows a unicellular structure. Okay, they are typically saprophytic, they are decomposers, they are parasitic, or they are corpophilous. The mycelium is branched and septate. Branched, they are completely showing up branches and they have a separation, then they are called as septate mycelium. Asexual spores are conidia produced on the conidiospores. Children, these are the asexual spore structure, these are the conidiophores. They are producing the spores called conidiospores. Conidia on germination produce mycelium. When the conidia germinate, they produce the mycelium. Ascospores, sexual spores, are produced into the ascus, arranged into a fruiting bodies called ascocarp. So, there are fruiting bodies which are called ascocarp. Ascocarp are producing spore called ascus, which basically led to the production of spores and the fusion of gametes through these spores. The examples of the ascomycetes are Aspergillus, Claviceps, Neurospora. Neurospora is used in the biochemical and genetic work. Yes, Neurospora crassa is used for the study of genetic material, the genetic that is used a lot. This is basically, you can also get Drosophila of plant kingdom. Okay. So, Drosophila of plant kingdom is also called the Neurospora crassa, which is basically used for the genetic study. They are used a lot in the genetics to study all the genes, their formation and everything. Belongs to the kingdom fungi class ascomycetes. Okay. So, these are the few features of the ascomycetes, that they are a sac fungi, they are multicellular, they are unicellular, which is yeast, they are saprophytic, they are decomposers, or they are corpophilous. They show the mycelium which is branched and septate. Asexual is through the conidiospores, bisexual through the fruiting bodies producing the ascus through the bodies called ascocarp. Okay. Children, the examples which are Aspergillus, Claviceps, Neurospora and the Neurospora crassa is all the used for the genetic makeup study, which is also called the Drosophila of the plant kingdom. Clear? So, this is about the ascomycetes, the features of ascomycetes. Next, we are going to talk about the basidiomycetes. Okay. Next is the basidiomycetes. So, children, as the name suggests, basidio, you should assume that they also have this phase, or basically a place which gives the generation to the spores, which is called basidiospores or basidiocarps, whatever you can say. The common forms are mushrooms and puffball. Mushrooms which are edible also, okay, they also belong to the class called basidiomycetes, which is the common forms are mushrooms and puffballs. They grow on the soil, tree, stumps, as well as act as a parasite. They can grow on the soil, they can grow on the tree stumps, they can also act as a parasite. Okay. The mycelium is completely branched and have a separation which is called septate. The asexual spores are not found. So, the basidiomycetes is not reproducing asexually because there are no asexual spores. Yes, there is a vegetative reproduction by fragmentation is common. Yes, they can reproduce if the fragmentation is done and each part develops into a new individual. If the body breaks down into few fragments and each part develops into a new individual. Okay. The sex organs are absent, but plasmogamy is formed. The complete sex organs are absent, but yet they show the plasmogamy. There is a dikaryotic phase occurs which gives rise to basidium arranged on the basidiocarps. Yes, a dikaryotic phase, it means there will be a two nucleated stage giving rise to a basidium arranged on the basidiocarps, which will going to produce basidiospores. Karyogamy and meiosis takes place in the basidium, producing 4 basidiospores. So, children, karyogamy is the fusion of nucleus, yes or no? This is the fusion of nucleus, and of course the meiosis, because meiosis is going to produce, if there is a dikaryon stage which will undergo meiosis will produce NN spores. Yes or no? Again these NN spores will form a 2N which is a dikaryon stage. This is how the karyogamy is taking place, and the meiosis is also taking place in the basidiomycetes. The example for the basidiomycetes is the agaricus, Astrelego and Puccinia. These are the few examples of the basidiomycetes. Okay, we have studied the phycomycetes, we have seen the characteristics of ascomycetes, we have seen the characters of basidiomycetes, and the last which came in the limelight is the deuteromycetes, which is also called fungi imperfecti. Why deuteromycetes are called fungi imperfecti? Because there is only asexual and vegetative phase of these fungi are known. It is called fungi imperfecti because no sexual reproduction. There is no sexual reproduction, and that is why deuteromycetes are called fungi imperfecti. Okay, they are known as fungi imperfecti because there are no sexual reproduction. Once perfect sexual reproduction was discovered, they were one moves to ascomycetes and basidiomycetes. If in case by any chance the sexual reproduction has been discovered in the coming time or whatever, they will not be existing in deuteromycetes. We will move it to basidiomycetes or ascomycetes often. Okay. So, children, deuteromycetes reproduce only by conidia, conidiospores. The mycelium is completely septate and branched. They are mostly decomposers and some are saprophytes or parasites. Yes, they can be decomposer, they are typically saprophytes and parasites. So, sometimes this do show some phase which is like a sexual reproduction phase, okay, and they are often moved to ascomycetes and basidiomycetes, okay, and if they completely show the sexual reproduction, they might even get into basidiomycetes or ascomycetes, but till now we have kept it inside the deuteromycetes only because they are completely called as completely show the absence of the sexual reproduction, which is why they are called as imperfect fungi or fungi imperfecta, whatever you wanted to call it. The examples of deuteromycetes is Alternaria, Colitrichum and Trichoderma. These are the few examples of the deuteromycetes. Clear? So, children, we have seen the habitat, we have seen the structure, we have seen how different classification has been done, we have seen the different classification and their different features, how they are different from each other, be it basidiomycetes, ascomycetes, deuteromycetes or phycomycetes. We have gone through all their features. Okay. So, children, this is what we are going to go through in today's lecture. Tomorrow we are going to discuss about the sexual reproduction or the reproduction in the fungi. Okay. So, today this is all about the classification and the whole habitat and the few general features of the fungi. Let us see what we have next. We have the other question. So, children, the mushrooms can be edible, yet they can be non-edible, and if they are non-edible, they are completely poisonous. So, the deadliest mushroom known as is, can you please answer? The deadliest mushroom known as is, yes, the deadliest mushroom known as is the Amanita. Amanita is the deadliest mushroom. If you consume it, it will basically create a poison inside the body and might get taste of death. So, the genus Amanita contains 600 species including some of the most toxic mushrooms found worldwide as well as some are well edible species. The genus is responsible for 90% of fatalities during resulting in the mushroom poisoning. Okay. So, children, the poisonous mushroom is the Amanita. Yes, a very important question, a very basic question, that the fungal wall is chiefly made up of what? What makes a fungi put into a different kingdom only? It was the cell wall. Yes, what it is made up of? What it is made up of? Come on, quickly tell me what is the fungi cell wall is made up of? Yes, children, the fungi cell wall is made up of chitin. This is what I have been telling you, non-cellulosic polysaccharides, and the chitin is creating the fungi cell wall. Create. Moving forward, children, next question we have, main component of fungal cell wall is what? Cellulose, pectin, dextrin or chitin? Main component they are asking. Main component of the fungal cell wall is what? The main component of the fungal cell wall is what? Come on, quickly answer. Simple, children, it has to be very simple. Chitin is the main component, and non-cellulosic polysaccharide, not cellulosic, non-cellulosic polysaccharides. Okay. The next question which comes in the limelight is the dikaryon is formed when? How the dikaryon will be formed? I have told you, how the karyogamy is basically taking place, and then the meiosis. What do you think? How the dikaryon is basically taking place? How dikaryon is taking place, children? So, meiosis is arrested, 2 haploid cells do not fuse completely, cytoplasms do not fuse, none of the above. Yes, of course, children, the 2 haploid cells do not fuse completely, creating a dikaryotic stage. Okay. Next, a pair of associated with the unfused haploid cells. The examples of unicellular fungi, very common. Children, the example of unicellular fungi. The examples of unicellular fungi is the Saccharomyces, which is yeast, which is the only unicellular fungi existing, otherwise the fungi shows the multicellular behavior. Okay. The next question is the mode of nutrition is heterotrophic in what? Albugo, Biopyphyta, Eleutherox, which show the mode of nutrition heterotrophic. It has the basic mode of nutrition which is the heterotrophic. Eleutherox, Albugo, Bryophyta or Pteridophyta. Children, the mode of nutrition is heterotrophic in fungi, of course, which is the albugo. Albugo is the fungi which shows the heterotrophic nutrition belonging to the Phycomycetes. Albugo belong to the Phycomycetes, show the heterotrophic nutrition. Okay. Moving forward, let we have the another question. The fungi differ from the slime molds in? How do you think the fungi differs from the slime mold? The fungi differ from the slime molds. Flagellated spores, zygospore, ascospore, basidiospores. What do you think the slime molds have and fungi do not have? So, children, the slime molds possess the flagellated spores. The fungi differ from slime molds because they lack flagellated spores. Yes or no? They have flagella in their spores. Okay. Next, what is the fungi imperfecti? Very simple question. What is the fungi imperfecti? What is the fungi imperfecti? Phycomycetes, Ascomycetes, Basidiomycetes or Deuteromycetes? Very simple. The fungi imperfecta is that which does not show the sexual reproduction, and the only fungi which does not show the sexual reproduction is the Deuteromycetes, and that is why it is called as fungi imperfecta. Okay. So, children, we have come to our end of the session. We have discussed about the whole fungi classification, their basic feature, their habitat, where they belong to, their nutrition, mode of nutrition, and how different classification, different classes possess different characteristics. So, the member of fungi are completely diversified in nature, they are cosmopolitan in nature, they basically present everywhere, they have asexual as well as the sexual reproduction. The Phycomycetes, Ascomycetes, Basidiomycetes, Deuteromycetes are further classification of the fungi, some which show the septate mycelium, the other show the aseptate mycelium, which is kind of a primitive kind of a or a lower fungi. Okay. The only fungi which does not show the sexual reproduction belong to the class Deuteromycetes. They are also called as fungi imperfecti. The mode of nutrition is typically heterotrophic, which could be parasitic, which could be saprophytic, and the most special one is the symbiotic association shown by the mycorrhiza or the lichen. So this is all about today's session, children. Tomorrow we're going to discuss about the reproduction in fungi. Do not miss that class. We're going to discuss a detailed discussion of the life cycles exists by different class, and we're going to discuss in detail about the reproduction taking place in the fungi. So children, at the same time we'll meet, but before you go, let me introduce you to this. This is the Embibe Lens app. So what you have to do, children, you don't just have to download this app, scan the different text you wanted to, it will highlight the keyword and you can see everything in 3D. So please go download it, have a very good experience of visual learning, and do tell me how you found it out. Okay, and the next, children, you can join us on the Telegram channel. This is the Embibe app. You can log in using the phone number or the email ID. The OTP will be generated and you can basically get through the whole. There will be a home page, learn page. There's a home page, learn page, and there is a test and achieve. So on the home page, if you press, there is a whole what you called as a live Embibe live session. All the respective classes will be present over there, so you can also register yourself. All the classes are present on the Embibe app. Do join us on the Embibe app, because different experts are teaching you different subjects having in the sequence. I'm going to discuss all the important stuff you wanted. Okay, and children, you can also join us on the, this is our application which you can download from the app store and wherever you want. We will meet again tomorrow at the same timing discussing about the reproduction, the fungi. Till then, children, please take care, keep imbibing. Bye bye.
Keep imbibing. We believe in you.